Just while we're waiting for Dave, I can save a little bit of time for him by doing a little bit of his housekeeping. So obviously, I think some of you would have been with us for the first set of nodes, the 5 that we did a couple of weeks ago. So that went really, really well. Dave will have to be quite a tight time master again this morning to make sure that we have time for all of the node presentations. We should have a little bit of time at the end of each, after each slide presentation for any immediate questions. So what we did last time is I think people put those in the chat as we were going through. So we tried to kind of review them as we went along, and then pull out a couple to ask the speaker or speakers straight after their note presentation. Then we've also got a nice bit of time at the end where we should be able to do a little bit of Q&A and discussion together on any common strands that come out. Because we have 6's time, we probably won't have quite as much time at the end to do that, the cross node discussion. But as we say, we'll stick tightly to time so that we should have some space there. And also, again, looking at our participants for people who weren't able to join last time, I think, Nikita, is the first presentation or the first video, is that already available on YouTube for people to watch? Was that going to be?
Not yet. So we're just going to put up both the sessions together.
Great. Okay.
Yeah.
Super. So do have a look at that if you weren't able to join the first ones, because I think together the picture across the 11 nodes, both in terms of the more disease-oriented and also the really cross-cutting ones, altogether I think it comes out as a really interesting picture, and there's lots of kind of common themes that we're picking up. And it's great to have a mixture, as we did last time, of people from our nodes, but also members of our PAG, that's our Platform Advisory Group. So we have a really diverse group, and for that reason, I know you've all had the message from Nikita, node leads, but we ask that you try to keep it not too scientific and explain, jargon wherever you can.
Vicky, thank you for standing in. Unfortunately, my office has been turned into a film studio, and the bad news is they're not even filming me. So I've been thrown out. So I'm not entirely convinced that I won't be thrown out from where I've perched. So that's me told. So welcome, everybody. I know Vicky's said that. This is the second of the node showcase sessions. I think, as Vicky was saying, the first one was a really brilliant discussion. Lots of themes came out that people have been following up on. I think some sort of real sense now that people are up and running of the opportunities that are offered by collaborating across, and in particular that there are the two most cross-cutting nodes in methodology and in trial methodology and patient focusing activity are relevant to everybody. So when that video does go up, I would recommend that people have a look at it. So we have quite a packed agenda today because we have 6 of the nodes showcasing themselves. So if we could keep to time, we do have some time left over at the end for questions, and I suspect what we'll do is do Q&A as we go along, and that will eat into the time. But we do have 90 slides in the deck, I think, so it's going to be a bit of a canter. So, but the question and answer bits last time were brilliant and really added to the value of it. So we're very keen to be able to see that again. So I hope all that makes sense. If I do get suddenly kicked out from here, Vicky will pick it up and I'll go and find somewhere else that I can go and perch. That's me told in the organisation. So the first of the nodes this morning is the Cardiovascular Initiative. It's James Ware. Thank you, James. And as I said, welcome everybody to this.
Hi, thank you. Can you see and hear me okay? Yeah, I can see some nods. Fantastic. Okay, so if you want to jump onto the next slide, I'll keep it, try and keep moving. So I thought I'd just start by showing you who's who briefly. Kate, the people on the top are the other 3 co-applicants. on this. Kate Thompson is one of the senior lab scientists in Oxford in the genomic lab hub there. Tom Lumbers, she's not on the call today, I'm afraid. Tom is on the call. He's a cardiologist by background, expertise in genetic epidemiology and in electronic health records as well. Some of you may have met Tom as the lead of the EHR GSIP at Genomics England. And Kathy Sadlow, also not here today, was the Chief Scientific Officer at UK Biobank. You may have met her there, and heads up the British Heart, well, did head up the British Heart Foundation Data Science Centre, which is part of HDR UK, but has been seconded to a big longitudinal study elsewhere now. And then over on the left, a couple of key people. Claire Stockwell is coordinating our node and is also on the call. And Rachel is in my team and has been managing some of the projects. So next slide, please. Thanks, and another one, thank you. So I thought I'd just very briefly say a little bit about the rare cardiovascular diseases we're interested in. I won't go into this in too much detail. These are big health burdens. They come to public attention often when, in the context of sudden death in young athletes is the kind of thing where the reason why the public encounter these conditions. Actually, there are a number of conditions that are really not that rare. Violate the kind of some of the rare disease definitions if you just view them as hypertrophic cardiomyopathy or dilated cardiomyopathy or familial hypercholesterolemia, for example. But when you start digging into any particular genetic subtype, you immediately become rare. And obviously, as we need to be precise in our treatments and diagnostics, we need to apply rare disease approaches. But it does mean that we have sort of cohort sizes and numbers that make certain sort of statistical things tractable that might be more challenging in other areas. I thought it's helpful to say that the commonest of these typically present in adulthood, actually. So they're usually autosomal dominant, inherited. They're very genetically heterogeneous. So typically, well, not very, tens of genes can cause each of the main phenotypes that we deal with. And within those, there's a lot of allelic heterogeneity as well. One of the key things is they have incomplete and age-related penetrance. So as I said, they're adult onset diseases. There are lots of people carrying predispositions who never manifest. That's really important because when we find a patient and we start cascading out to families, it's very difficult to make a yes/no decision in a family member. Essentially, most family members end up in long-term surveillance because you can't rule out that they're going to get one of these diseases later in life. So they're big disease burdens. And the node is mainly focusing on the cardiomyopathies as the sort of most prevalent conditions. Okay, if we jump on to the next slide, please. Oh, and just advance, I think that it looks like there's an animation on there that I should have taken up. So just to say that these are kind of, I'm afraid, everyone's problem, even if you're not interested in cardiovascular disease, because they appear on the ACMG list, for example. About half of the genes that the American College recommends we should report secondary findings in are associated with inherited cardiac conditions. You know, companies like 23andMe are offering reports returning these sudden death risk genes, even though we really have very little understanding of what the actual risks are to patients found to carry variants in these genes when they're picked up incidentally. So that's a little bit of context. If you advance again. And one more, thank you. So I thought I'd tell you what our plan A, or what we actually said we're going to do, is first in 3 slides. And then I'll just tell you a little bit about how that's evolved. So we laid out, we saw the main challenge in our area as being one of breaking down data silos, that essentially we need access to the nationwide pool of patients if we're going to find the 1% who have cardiomyopathy due to phospholamban or the cardiomyopathy due to a particular genotype for a study or a trial, and to understand the natural history of different genetic subtypes. And it's a very siloed landscape. So the first project that we defined to break those down was essentially to build a database of all the genetic testing results in the UK. So a lot of, there are only 4 labs carrying out cardiac sequencing, or 4 of the hubs. And it should be possible to get that data into one place. At the moment, lots of time wasted with phone calls between labs to say, have you seen this variant before that we haven't? And essentially, it's limiting high quality genetic interpretation. So we wanted to follow a path that's been forged by CanGene and CanVar, where genetic testing is submitted to NDRS, the National Disease Registration Service, who process it, collate it, and then publish kind of variant level information, having stripped out the patient level data, which supports variant interpretation. And we were then going to display that in Decipher, that many of you will know as a sort of rare disease, a platform well known in the clinical genetics community. And so that was our first plan, and that would be a link from each GLH into NDRS was the plan. And then we hoped that some of the individual level genetic data we would also be able to make available for research with an additional level of consent, but most of that activity would not be patient level. It wouldn't require consent. So if you could advance, please. The second project then was to try and establish a national cohort of patients with cardiovascular diseases with consent. And our plan was that we would consent people when they had their NHS genetic testing in a similar sort of way to Genomics England and the National Genome Research Library. And we planned to put the data that we would collect, as I said, from the genomic lab hubs, etc., into a research environment at the BHF Data Science Centre. And then if we advance one more, we were then, our third kind of flagship Pathfinder project was then to enrich some of the data on that cohort, particularly by adding common variant genotyping for polygenic risk scores and things which are looking very promising in some of these diseases. And that was going to be a Pathfinder. We also want to enrich the cohort with phenotype data, so pull cardiac MRR, ECG data, that sort of thing, into this research environment. So that is what we said we would do. If you could just advance. Of course, everything's changed, but not very much. The vision hasn't changed, but the way to do it, I think, has. And our key change is we have become, we plan, if we can pull this off, to lean much more heavily on NDRS. So rather than, for example, having, well, yes, so this has been introduced to everyone, but there are a few key things. One is that formally the scope of NDRS is confirmed rare disease, but also suspected rare disease, high genetic risk of rare disease, and family members of rare disease. So it's quite an encompassing scope. And you can see at the bottom, there's the definition of rarity, or you can have an OrphanNet designation, or in fact, anything that's in the National Genomic Test Directory is eligible for this data capture. And so this doesn't need consent for this data to be collected. So if we could jump forward. So what we would like to do is create registration records in NDRS for the cardiovascular patient community. And we thought we would start by catching everyone who has genetic testing. As I said, there are only 4 labs doing it. And so this was sort of in our original pathway, but we think that rather than connecting with the genomic lab hubs, we can actually connect to the PLCM patient level something monitoring. It's basically the financial returns for the lab hubs, which already submit that to NHS England. So we've had to jump through a few hoops, but we think we can basically get a list of everyone who has had genetic testing with just the highest level data of what the test was for and whether there was a pathogenic variant of VUS or no variant found, essentially, with a single point of data linkage basically into NHS England. And that has been signed off in principle, but there's some QC steps to go through before we can access that. And then I think once we've done that, and I feel we're very close to it, we then have a register of a sort for our disease area that should be capturing some, several thousand patients a year going through the testing pipeline. We then want to incrementally improve that. So the next level is to try and catch people with HES data coding for these conditions. The problem is that we're going to do some validation of that. The problem is that ICD-10 codes are limited in this area, and it's limited to people who have inpatient care, of course. So the next layer is to try and access secondary care records, letters and stuff. And so with one of the GMSA networks of excellence, we are doing some pilot work to try and be more comprehensive in the patients we catch, in addition to those having genetic testing contemporarily. We advance one more. So I'm sorry, this isn't very high quality. If you just, sorry, I was going to animate that. If you just jump back to so that we only see the core of this initially. On the left, so we have identification of patients from their genetic test in the first instance, later from hospital episode statistics, and that populates the middle box, which is a registry held in NDRS. We then revert to our original plans. So having established the existence of people who have genetic testing, we then actually capture the extra genetic test data, so capture the reportable variants into a database also in NDRS, and hopefully the VCF files that might be useful for various forms of research. Do the research that I previously defined as our first node project. Again, none of that needs consent, and the patients then go and the data's results are disseminated through Decipher. And then our cohort, again, we, rather than contacting people at the point of genetic testing, we contact them retrospectively once they're in this register. So that's been done before. For example, the Melody study is an exemplar of this, using Ipsos Mori to write to people on the basis of an NDRS record. And so we plan to have a similar model. We've already got a patient-facing e-consent portal. And we then plan to, as I said, build up that consented cohort and then mirror their data in an accessible, trusted research environment to the BHF Data Science Centre, and to perform the sort of various forms of data enrichment that I alluded to before. So I think that's probably all I've got time for. I think that's my last slide. Yes. I hope that was just about in 10 minutes and gives a bit of a flavour of what we're hoping to achieve.
James, thank you very much. It's really interesting. We do need to move on. So next it is Histionode with Matt Collin. Matt.
Great. Thanks, Dave. Good morning, everybody. Move to the next slide, please. Thank you. So our world is a macrophage. That's otherwise known as a histiocyte. And we have two diseases that actually have been linked together at several levels for many decades, even though they're slightly different in their pathology. But one is a group of rare cancers, so histiocytic neoplasms, and you can see their three-letter acronyms that describe them, LCH, ECD, those kind of things. They're super rare. They're sort of a few per million incidents. And then we have another set of disorders which are overactive macrophages, but the macrophages are driven by other problems with the immune system, so a kind of immune dysregulatory set of disorders. Traditionally, these are been divided into primary HLH, which affects young children and has high penetrance single gene mutations, and secondary HLH, which afflicts adults, usually often with an identifiable trigger, but probably on a genetic background. The incidence of that is one of our projects to define the disease burden, because the recognition of this HLH condition is a work in progress. It has improved considerably as a result of COVID causing hyperinflammation and as a result of related cytokine storms after CAR T therapy in cancer. But basically, one of our tasks is definitely improving awareness of these conditions and defining their epidemiology. Next slide, please. Slightly messy team slide, but we have diverse range of co-directors and co-Is who received funding, and a bunch of collaborators as well. You can see we outlined 3 networking objectives, including engagement. Biobanking is a big thing for us because our diseases sometimes occur acutely and make people very sick, and even a significant rate of mortality within a couple of days. So it's quite challenging to get in there and get samples to do meaningful research with. And then a couple of projects, 1 and 2 are focused on HLH because of this high mortality, particularly in children and young adults. And then project 3 is related to the histiocytic neoplasms, where we're interested in somatic mutations that cause these cancers, and how that can be used for diagnostic and prognostic information. There's one person missing from this slide. Apologies to Tom Ewan, who's our project manager from Newcastle, who's been absolutely superb and been a really steady hand keeping us making progress with the regulatory issues and things. And also Liz Jury at the top right is on the call, I saw, so I'm going to show one of her slides later. Next slide, please. So the first sort of networking activity we talked about was biobanking. I have to say it's been a little bit frustrating. we advertised that we had a registry, a research tissue bank already running for now 5 years, and it was one of our kind of assets for making the application. But being a research tissue bank is not eligible for the NHR portfolio. That's meant that basically it doesn't get looked at in any centres, and because we have these high-risk patients turning up all over the place, many different trusts, we don't have time to centralise the patients. We really need to open in multiple sites, and that's been very difficult to solve. The workaround currently is twofold. One is Jessica Manson, Liz Jury, Beth Payne at UCL have proceeded with REC approval of a project on HLH, and have got well underway with recruitment there. We have some recruitment to our UKHR in Newcastle because we recruit local patients over a while. But really, the only solution we found is to resubmit our whole node on block to REC as a sort of multi-stream research project, because that's the only way we're going to get open at multiple sites. And that's really delayed our first sort of networking activity, unfortunately. Next slide, please. A happier story is the NDRS story. I think these efforts were already ongoing when we started, and were highlighted at one of the previous platform meetings. So the current lead for this is Fiona Pearce, who's already embedded in NDRS, but also works at Nottingham University, and she has given us a good example here of how health data can be exploited, again, to really define the basic epidemiology of some of our conditions here, this in HLH, and also to showcase the ability to reach out to Europe. So this is a very large study that's ongoing, including data from 29 European countries. It's based on ICD codes, and simply asking how many people are dying of HLH, the hyperinflammatory condition. Next slide, please. And you can see on the top left here, really, I think this is the sort of first solid demonstration that this really is a primary and secondary disorder. You can see deaths per 100,000 occurring in neonates who have primary HLH, and then in older patients there's a big secondary wave, but there is a sort of continuum, and there's likely a crossover between germline and somatic genetics playing a role here. The other interesting observation is that the death rate, the mortality, the standardized mortality rate, appears to have increased in the last decade. But this is almost certainly because the disease is being better recognized, which is good news, but it makes slightly troubling reading when you first see this picture. The kind of correlative evidence for this is that the countries in Europe mapped at the bottom there that have the highest mortality rate also happen to be, oh, sorry, go back one, hadn't quite finished. They also happen to be the ones that are publishing the most on HLH. So it appears to be an ascertainment bias here. Anyway, so lots of good news there, but still plenty more to do. The next slide is from Liz Jury and Jessica Manson at UCL, and they have very successfully launched their project using a sort of multi-omic approach to analyze fresh samples of patients with HLH. You can see the summary of the workflow here. It's a very complex project, but there are clear goals that will come out of this in terms of understanding the pathogenesis, identifying biomarkers, because key thing for HLH is early diagnosis, and then beginning to look at disease endotypes, whether it's a different genetic background or trigger, or something that means that there's a different flavor of the TMI, the too much inflammation, which then might lead to specific precision medicine treatments. So if there's any questions about this, Liz is on the call. I'm sure she'll be happy to answer those. Next slide, please. And then I just wanted to jump to Project 3, which is back to the neoplastic histiocytosis. So this is Langerhans cell histiocytosis, a rare blood cancer of children mainly. And this is our attempt to exploit everything that we can about cell-free DNA. So the so-called liquid biopsy, the presence of the mutation that drives the disease just in a peripheral blood sample. It's a slightly busy slide, but basically Langerhans histiocytosis has a number of risk strata, and each of those figures, it goes low to high, left to right across the panel. And what you see in panel A is that the black part of the pie charts are the ability to diagnose a somatic mutation in a tumor lesion just from the peripheral blood. And this is actually a very sensitive test that might save children a biopsy. Certainly all the high-risk patients, the MSRO pos on the right-hand side of that panel, they can all be diagnosed from the blood. The level of the mutation correlates with the risk of the disease at presentation, so that has a potential to add to clinical risk stratification. And C, on the right-hand side, you can see those survival curves. The level of mutation is also giving us a strong indication of the event-free survival. So this kind of project is only possible with big national and international collaboration, and thanks to the funding, we're really able to hold our end up in the UK. Of the 270 patients, we contributed 100 cases to the study and about another 100 from Italy. So it's really a significant contribution from us. Next slide, please. So PPIE, HLH side of the project really leading the way with this. They have a very active regular meeting chaired by a clinical nurse specialist. We're also all recording little videos for our PPIE partner Histio UK, which deals with both sides of the disease. That's in progress, and a bunch of us are going to the Histiocyte Society annual meeting, which is the global meeting for histiocytosis in a few weeks' time. Next slide. So I was asked to say challenges. Again, the portfolio issue is a bit depressing. I would have hoped that could have been fixed at a high level. We're still struggling from the fact that we're basically doing observational studies with low volume, high risk disease, but we're being judged by the same standards in terms of risk aversion as a clinical trial. And obviously everyone's a little bit impaired by the overburdened NHS and the lack of commissioning for some of the treatments that we find evidence for. Next slide. Final slide. Biggest impact. Well, I really like the way our clinical advice networks work. It helps everyone engage and feed patients in. I think we're making progress with digital health data. It's very cool to hear that might get even easier. And I think reaching beyond the UK is important. In terms of science, we have a platform that is going to disentangle germline and somatic genetics across both of these diseases. And yeah, hopefully, sorry, I'm going on about the portfolio, but that's my final point. I'll stop there. Thanks very much.
Thanks, Matt. Very interesting. We should probably move on. So the next one is UPNAT, which is Alice Davidson and Nick Lench. Alice and Nick.
Hi.
I don't know who's starting.
Can everyone hear me okay?
Yes.
Yeah, brilliant. Okay, so if we skip to the next slide. So it's my pleasure today to introduce UPNAT and give you a brief overview of what we're all about. So Hyan Jo, our node director, can't be on the call today. She's at an international meeting, but I'm here. As we've heard, Nick Lench is also here. Our node director, Catherine Ryan, who many of you may already know, is on the call, and our newly appointed node research associate, Ellie Whittle. So you have hopefully a nice flavour of the team here today. So as our node name suggests, we're all about a platform technology. Really, we have the central goal to provide the UK with a roadmap to accelerate the translation of nucleic acid therapies for rare diseases in the UK and beyond. And we are hoping to be able to do this by bringing together the relevant stakeholders, so the scientists, the clinicians, industry, and of course, regulators and patient advocacy groups, into a much more streamlined and national network to promote skill sharing, innovation, and development in this preclinical translational space. So the node itself is hosted by UCL, but we've also got very crucial engagement from academics at the University of Oxford, Cambridge, NATA, key input from Genetic Alliance and Genomics England, as well as our integrated BRCs. And we've built this node around 3 different work packages, which I will briefly introduce you to today. So if we could skip to the next slide. Work package 1 is being led by Professor Mina Wrighton, who's a clinical geneticist and director of the Cambridge DRI, alongside myself. I'm based at UCL Institute of Ophthalmology, and my background is in molecular genetics and cell biology. So really, the overriding goal of this work package is to develop guidelines of tools and resources to promote NAT strategy design and development in the preclinical space for rare disease. And so far, our efforts in this first year have been really centered upon establishing working groups to identify these key 4 areas. So the variant themselves, the genes in which variants occur, the diseases that are affected, and the patient groups themselves. So leading experts in these areas to develop a set of guidelines to facilitate prioritization of variants, genes, diseases, and patients that would benefit from nucleic acid therapies. And by doing this, we've also been keeping a keen interest in the international space and development in this area. And if we go to the next slide now, please. This has naturally really nicely led to us being invited to partake in an N=1 led initiative to develop some criteria around variant selection for nucleic acid therapy development. And we've contributed to the development of these guidelines, as I've said, that have been led by this international collaborative group. And this has now led to a preprint and a submission of a paper, as well as some online materials and a variant prioritization tool. So that's a really nice outcome so far. But if we skip to the next slide, I think through the engagement of us in this N=1 led initiative, it's really enabled us to take what we'd originally envisaged for this work package and adapt and shape to now this update. It's a really fast moving field, and we're really mindful of wanting to be able to develop and facilitate these guidelines that are actually going to work in a UK setting. And this variant focused approach has been fabulous, but it's also really highlighted to us that we also have these other key areas are so crucial. And these are very much what our working groups are now going to focus on. So the gene, disease, and patient selection criteria we need to get in place. So if we skip to the next slide. So really, just to sum up for work package 1, we're aiming to develop an evidence-based approach to identify NAT targets within the UK system. And by doing so, we're hoping to create this expert-led framework that's going to be really adaptable and malleable as technology advances in this space. We hope to be able to publish proof of concept guidelines within about a year to a year and a half that we can share with the community. And in the later stages of this funding, we hope to be able to trial these guidelines in very select services, highly specialized services, to see how they actually play out and what how their clinical utility looks. So if we jump to the next slide now, we've got Nick here on the call, who's leading work package 2.
Great, thanks, Alice. Yeah, so as part of this process, we've actually identified a patient at Great Ormond Street, working with clinicians there and UCL Institute of Child Health, that we're going to use as an exemplar. So this patient has a splice, an deep intronic splice mutation, splice site mutation, and we've developed a splice switching antisense oligo to correct this defect. But of course, it's all well and good doing a lab-based system, but actually if we want to scale this up, if we want to move this into clinical development, then how do we, how can we find the capability and the capacity, the resources in the UK to manufacture these antisense oligos, to scale them, and to also look at the tolerability and toxicology of these particular agents. So this is what work package 2 is focused on. So at NATA we have the ability to do some scale up synthesis, but we are not a GMP facility. So we are now working with CPI, the Centre for Process Innovation. They are part of the Catapult network, the Manufacturing Medicines Catapult, and they have funding to set up a oligonucleotide manufacturing innovation centre of excellence up in Scotland. So we'll work with CPI to really develop a protocol for how we can scale these or the synthesis of these particular oligonucleotides. And then in terms of the tolerability and the tox, again, we can do some basic tolerability studies in rodents here at MRC Harwell. But again, we're looking to work with CROs such as Charles River, who are already developing NF1 therapies, have a lot of experience in this area, really, again, to work out how we develop those protocols that we can then take to regulatory authorities such as MHRA to show that we have a standardized protocol for moving these therapies forward. So the good news is that most of these antisense have a standard chemical modification for which there is already a lot of safety data. They're small molecules, they're not biologics. So again, that provides us with I think quite a lot of advantages. So this hopefully will act as an example. We'll have generate data through work package 2, and then as new patients are identified and selected through work package 1, then obviously the challenge then is to identify ways to fund the clinical development and translation of those therapies. So I'll hand back to Alice for work package 3. Thank you.
Thanks, Nick. So we've also got a third package that's running in parallel, and this is led by Professors Francesco Monotoni, who's based at UCL, and Carlo Rinaldi, based in Oxford. And the key goal of this third work package is to build this multidisciplinary forum to exchange information on NAT clinical trial regulatory approval processes between key stakeholders. So I should say Francesco and Carlo have got a wealth of experience in this space. Yeah, really, they're looking to improve communication between often these very siloed groups that tend to work on rare diseases, and ensure that we're really learning from experience in the rare disease field. They've already made some really great progress. So they're in the process of setting up a registry of adverse events observed in NAT investigator-initiated studies, which is really promising. They've also got plans to develop a white paper on preclinical requirements for regulatory approvals in this space, and they are already in continual dialogue with the regulators around this topic. So it's going to be exciting to see this move forward. If we jump to the next slide. So just to highlight, for us this year was an inaugural symposium that we held back in June. This was spearheaded by our node manager, Catherine Ryan, who was amazing at bringing us all together. We had a fantastic lineup of speakers, and we got loads of engagement across the key stakeholders we're looking to really bring into the community. And we had lots of panel discussions as part of this day. So it gave us, as PIs on this award, really excellent feedback at the initial stages to think about how we want to shape the work packages moving forward. So that was an exciting development, and our plans are to continue to have this format every year as we move forward. If we jump to the next slide. I just wanted to end on potentially some of the challenges that we faced in this first year. I think a lot of these challenges are going to be shared by many of us on the call. So we absolutely knew that at the outset of this call, knowledge exchange was the crucial kind of nut we were trying to crack. We're all in the rare disease space, working in our silos, getting on, doing great stuff. But actually, by sharing information and bringing all these key partners together, we will be so much stronger. So we've always got this at the forefront of our mind, and we're trying to come up with new and innovative ways to promote knowledge exchange. But nonetheless, it's a challenge, and it's something we're continually having to push ourselves to do better at. Data access, I'm not going to spend too much time talking on that because we've already touched on that quite a bit, but clearly working with rare genetic diseases, wanting access to big data series, there's a huge amount of legislation and issues around that, and that's not unique to our node. We're really mindful of legacy as well. So we want to ensure that the work we're doing now is actually going to be useful and have clinical utility beyond this initial 5-year period of funding. So keeping that in mind, I think that is a key challenge we have. And we're also very mindful of needing to ensure that we position UPNAT in an effective way, because it's amazing we have this funding, but really to make and accelerate progress in this space, we need to position UPNAT in a way that we can leverage further funding, both within the UK and internationally, to accelerate the development of nucleic acid therapies for rare disease. So I'll just end then with the last slide, which is kind of trying to sum up where we see how our UPNAT fits. So we want to be this integral linchpin in the UK, bringing together all these different really important stakeholders that will enable the translation of nucleic acid therapies for rare disease. But we know that we don't want to be UK centric, and we learn so much from the international community. So we're really mindful as well of ensuring, and I think Hyan is doing an excellent job of leading in this space, of really making sure that we're mindful of all these other international processes and pathways that are up and running, to make sure we're at the top of our game and really accelerating progress in this space. If we jump to the last slide, I'd just like to bring up the acknowledgement. So these are all the partners and institutions that have been involved with the node to date. I hope this list is going to continue to grow as we move through this 5-year period. And I'm really happy, along with my colleagues, to take any questions.
Thank you very much. Really interesting. So in the interest of time, we're keeping almost to time ish. Bad sharing. Right, so the next one is Joe Bateman talking about mTOR. And Joe, either now or maybe at the end, depending on time, you put a comment in about your experience about ICD-10. And I think it might just be useful to expand on that. Maybe we'll save that until the end when we do a sort of broader Q&A. But
okay.
Joe Bateman.
Okay, thanks. Great, thanks, Dave. And yeah, so move to the next slide, great. So our team on the mTOR node is, I'm obviously the lead, and then Laura is deputy director, deputy lead, and then Deb, who are both epilepsy clinicians at King's. And then at St George's, Francis Elmsley is a co-I, and we've got a really good PPI lead in Charlotte Ty at King's. And then through the grant, we've been able to recruit Sylvine, who's our project manager, who you heard from earlier, and Layla, who is a research assistant. So next slide, please. So yeah, this, just to give you a reminder, the aim of our node is to try to look at these group of rare diseases, which are preventive pathway diseases, and to bring together the clinicians, the scientists, researchers, patient organisations who are all interested in this group, about 14 different rare diseases. And underlying these diseases is a common molecular mechanism, which is known as the mTOR pathway. So we wanted to bring everybody together, and we got a good start quite early on, about a year ago, where we had a kick-off event at King's, and we brought together everybody that's involved in the application to get to know each other and to talk about the aims of the node. And apart from having lots of good discussions, we had a couple of nice outcomes from that, which was there was consensus to start some task and finish groups to look at some of the issues we discussed, and also a consensus to write and put together a position paper, review article to describe this group of diseases, because they haven't been brought together in this way before and haven't been thought of as a group of rare diseases before. So we also had a annual symposium in April, which we hosted jointly with the MRC Centre for Development and Neurodevelopmental Disorders in April, and we got a really good attendance level, and mostly science talks and clinical talks. We had a discussion at the end. We also had a talk from a patient with one of the diseases in this group, tuberous sclerosis complex. So that was very nice and went well. So we've now built up a kind of core of about 40 network members, and we've got a mailing list of over 100 members, and we have a newsletter put out about a week ago, and we do that regularly as well. The next slide, please. So in terms of enabling science projects, the first one is to develop a registry, including some of your nodes, to build these group diseases through the end, working with the MDRX. Bring up the next. And then the second aim is more, thank you. The second aim is more about the underlying mechanisms. So actually doing the bench science and looking at getting tissue from mTOR pathway patients and doing phosphoproteomics together with a collaborator at QUIT to look at targets of the mTOR pathway in these patient tissues. And then the second part, the second aim is to get blood from mTOR pathway disease patients and isolate peripheral blood mononuclear cells to develop into a cerebral organoid model for the most common disease in the group, which is tuberous sclerosis complex. And we're doing that in partnership with a company in Oxford, Stem Tech. And the third aim is then to try to develop non-invasive genetic testing for some of the really difficult mosaic diseases in the group, things like photophilic dysplasia, where there isn't a germline mutation, it's a somatic mutation. And at the moment, the only way to genotype the patients is to use surgical tissue. So I'll tell you about where we are with those. Can you move to the next slide? So for aim 1, we recruited Layla in November last year. So she's a research assistant, and primarily she's working together with the MDRS. So as we were asked to talk about challenges, as the Cardionode found, it's been a slow process working with the MDRS, but we did actually manage to get an honorary contract and a data access contract for Layla approved in May. So Layla is officially an honorary employee of the MDRS and is able to have access to their data systems, which is fantastic. And she's been working really well with a new colleague at the MDRS. And the idea then is to work with clinicians and the clinics representing the different diseases in the mTOR pathway, and to try to import data from those patients into MDRS. Next slide, please. So as Karen alluded to already, I borrowed this slide from Karen, so I can't take credit for it. But as Karen mentioned there, in addition to the cancer register and the NCARDS register, MDRS have recently started this government-mandated rare disease register to cover the majority of rare diseases which aren't included in the cancer and the NCARDS register. So we're really excited to be working with Karen and with her colleagues and through Layla to start to do that for the diseases that we're involved in. Next slide. So aim 2 is about primarily firstly getting the tissue from the patients to do the phosphoproteomics and to develop the iPSC organoid models. And again, in common with some of the other nodes, we having to get ethics approval for that as a new project. So Sylvine, who is our project manager who we recruited, has been really leading on this and doing the ethics application, putting it together. And we went through a number of different versions of this. We had one version that had a tissue bank, but as is mentioned, we've come up with the same problems that we can't get an NIHR portfolio approval for that. So we really streamlined and simplified our first ethics application to focus on getting blood from TSC patients. And although, again, that was delayed and just communications have been very difficult internally here, we have managed to submit it to IRAS, and we have our REC meeting actually tomorrow on that. So fingers crossed, if that goes well, then we'll be looking at patient recruitment in early next year. Next slide. So aim 3 was planned to start a bit later than the other aims, and we have made some progress. This actually, Laura had an already existing ethics approval to recruit patients as part of one of her studies. So we put in an amendment to expand the number of NHS sites that we can recruit from, and that was approved, which is great. So research into that part of that aim will hopefully get started middle of next year. Next slide. PPI has been a big part of our project, and we really got that going early. Charlotte's been really great leading on that. So we had an online PPI workshop for 9 different patient organisations that represent the different diseases in this group, and that went well in December. We've recruited a PPI panel of 6 patients or patient representatives in July, and they've been very busy helping us in various looking at the ethics application, giving us feedback on that, on the manuscript that we had in preparation, and we've also got an in-person PPI workshop planned for December. Next slide. I just wanted to mention things that weren't planned in the grant application and have kind of come out as it had evolved really as part of the work that we're doing and the collaborations that we have. So the first is a new NHS clinic at King's College Hospital that's being led by Deb, who's responsible for pediatric mTOR pathway patients, and Laura, who sees the adult epilepsy for mTOR pathway disease patients. And this is a new clinic that started very recently. And the idea is to try to funnel the sort of ultra rare mTOR pathway disease patients who have epilepsy to King's to see specialists here who know all about those diseases, and potentially also get them as part of N-of-1 trials that we'd like to get going in this group. Then we've also had some successful funding. Can you go back? So we had partners on an Innovate UK grant with Oxford STEMtech and a Swiss company, mTOR Biosystems, to use the iPSCs that we're going to generate through the node. And then we've got a PhD student starting October next year, who again will work on iPSCs. And we have a pilot grant with a company called Cosine Therapeutics to look at repurposing mTOR pathway inhibitors in mouse models, which is ongoing. So last slide. I just thought I'd leave you with the poster that Laura put together for the clinic that she started as an advertisement more than anything else. If anybody comes across a patient with a mutation of one of these genes listed in the box below, and that patient has epilepsy, then please get in touch with Laura and Deb, because we'd be very happy to include them in this new connection. So that's all I have to say. Thank you for listening, and happy to take any questions.
Okay, thank you very much. So next is lipidomics, and this really is Bill Griffiths. Apologies for that. I was getting ahead of myself. So Bill.
Okay, thanks very much. Okay, so maybe you can just go back to the last slide. So yeah, the title of our node is Lipidomics and Metabolomics for Rare Disease Diagnosis, and our core players really are the Swansea University, Cardiff, UCL and Manchester, and our clinical centres are University Hospital Wales, St Mary's Hospital Manchester, and Great Ormond Street. Okay, next slide, please. So the key members of our team. So on the left, you've got my colleague Yutin Wong from Swansea, then Graham Shortland and Stuart Moat in Cardiff, Mohsin Ali Asghari, our project manager. And then at the bottom, we've got Simon Jones and Andrew Morris from Manchester, and Kevin Mills from UCL. I've got one picture missing. That is of Owen Pickerel, who runs the Swansea Biobank that we're making use of. Okay, next slide, please. So really, our research challenge in this node is to get patients with a rare disease a diagnosis faster. And we want to try and do this by exploiting lipidomics and metabolomics technologies that are currently available in the research laboratories in the UK, but are not available to the NHS. So just a word about lipidomics and metabolomics. So in lipidomics, we measure all the lipids in a system, for example, a plasma sample. In metabolomics, we measure all the metabolites in a system. So in real life, we don't actually measure all, but actually in a single analysis, we do measure several hundreds. So basically the lipidomics and metabolomics, we're getting data for many different analytes in a single analysis. And this is useful in rare disease studies, as many rare diseases present with unusual patterns of metabolites. So we can go some way to helping in the diagnostics pathway. So that's the sort of first part of our project. We're also ultimately what we want to do is translate the methods that we're developing in the university labs ultimately into the NHS metabolic lab. So the methods of metabolomics and lipidomics ultimately are embedded in NHS laboratories. Okay, next slide, please. So we had our, the project started in June last year, and then we had our kickoff meeting in December, where we had a number of presentations and then lab tours. So here I'm actually chatting to Eluned Morgan, who's the first Minister of Wales, who came to our kickoff meeting, and I'm sort of trying to explain to her how lipidomics works. So if you take a look at the slide, so on the top left-hand side, we've got what's labeled LC, that's liquid chromatography. So we take our sample, usually a plasma sample, and put it onto our LC instrument. This separates out the different metabolites. They then go into that big box, and that's the mass spectrometer. And the mass spectrometer identifies the different molecules and determines their abundance. And then we get a readout on the screen. And as I mentioned it in the last slide, so some rare diseases give very unusual patterns of metabolites. So then by looking at the readout, we're able to go some way to identifying different rare diseases. Okay, next slide, please. So our fundamental challenge, right, is get this rare diagnosis faster. So we started with the low-hanging fruit. So the low-hanging fruit really is the variants of uncertain significance. So if there's a variant of uncertain significance in a gene coding an enzyme, and that variant is pathogenic, then the enzyme will be defective. So if the enzyme is defective, there'll be a buildup of the substrates of the enzyme and a loss of products. So this will change the metabolic pattern, something we can measure and something we can use to help make a diagnosis. So as I said, we've been doing this for a number of rare diseases, and we've confirmed or otherwise the significance of the variants of unknown significance for some of the diseases. Examples are CTX, SPG5, and Niemann-Pick. So these happen to be rare diseases that we're particularly interested in. Another tougher challenge that we're working on is diseases which where there is no diagnosis at all. So these what we might call syndromes without a name. And this is, I must say, this is a lot more tricky than the variants of unknown significance. But we're making positive progress. Okay, next slide, please. So ultimately, we want to translate our lipidomics and metabolomic methods from the research labs into the NHS labs. So we've been working with the instrument manufacturing companies, Shimadzu and Thermo, to translate our methods that we've developed in Swansea and UCL onto the actual instruments that are used in NHS labs. So we'll be at least be in a position where we've got everything translated that can move into NHS labs ultimately. Next slide, please. Okay, network. So I mentioned our core partners, Swansea, Cardiff, London, Manchester, clinical partners, University Hospital Wales, St Mary's, and Great Ormond Street. So we've expanded our clinical collaborators quite a lot, really. So now we've got on board Sheffield Children's Hospital, St Luke's Hospital, Bradford, UCL Hospital, Guy's and St Thomas and St George's, and they're all sending us now interesting rare disease samples to try and help make a diagnosis with. And we're also working with a number of patient organisations. So we've been attending meetings and making presentations. So example, Genomics Cafe Wales, which is run by Genetic Alliance, Alex, the leukodystrophy charity, Huntington's Disease Association, Motor Neuron Disease Association, Cure DHDDS, CamRare, and CTX Alliance. So we've been going around doing quite a lot of leg work, attending meetings, and telling people what we're up to. Next slide, please. Other achievements. Well, one other thing that metabolomics and lipidomics actually is really quite good for, it's monitoring the biochemical response to therapy, because in metabolomics and lipidomics we're really monitoring biochemistry. So we've been able to help in a couple of studies where NHS labs didn't have the capacity to analyze the metabolites that we can look at, and we've been monitoring response to therapies. We've got some grants, so we were able to get a MRC grant to investigate biomarkers for motor neuron disease, and we've also got another grant now to look at Huntington's disease. We've supported a collaborator in Sheffield to get an NIHR PCAF fellowship, and we've been making a lot of presentations also at scientific conferences. So UK MadLab, British Inherited Metabolic Disease Group, Rare Disease Implementation Network Wales, SLOS Scientific Conference, Society for the Study of Inborn Errors of Metabolism, and others. And another thing that we've now been able to introduce here is in Swansea is lectures on rare diseases to our undergraduates, into undergraduates studying biomedicine. So that's a new thing we've managed to do here. Okay, next slide, please. So impacts and challenges. So I think certainly with the variants of uncertain significance, we've had success. So this has allowed us to confirm the pathogenesis of a variant and allow them patients to go on and get treatments. Again, I re-emphasizing the monitoring the biochemical response to therapy, that works very nice with metabolomics and lipidomics. And then finally, challenges. So where we've got research nurses in place, then collecting samples and getting consent works very easily. But it's a lot more difficult in the other centres where we don't have a research nurse, really, because the clinical staff are really busy and they don't always have time to consent the patients for us. And what's ultimately going to be a challenge is actually making sure that these new omics are reproducible across multiple sites, so the methodologies really need to be very, very strong. So we have reproducibility, Swansea, Aberystwyth, Newcastle, Leeds, so we all get the same results. Okay, I think that's more or less it. So I'm happy to answer questions.
Thank you very much. That's very interesting. And last is, but by no means least, is John Sayer, my colleague from Newcastle, and CILIAREN. John.
Yeah, hi everyone. Thanks for staying on the call till the end. So CILIAREN is our rare renal ciliopathies national network. We actually changed the acronym at the advice of the MRC with something with vowels in it rather than RCNN, which was initially posted. But I'd just like to say that it's been a real joy actually to start this project, and I know I've maybe said some negative things about end cards and stuff, but actually it's all coming together very nicely, and it's a joy to be part of both the node and the hub. So next slide, please. So yeah, I don't need to dwell on this, but actually I think the reason I put this up is there's good synergies actually between CILIAREN and the lower urinary tract node, and also the mTOR node, and also nucleic acids. And so I think, you know, these interactions when we wrote them and when the grant process was underway were slightly aspirational, but I think now that it's time to realize them. And I think maybe this is one of the benefits of these meetings coming together, is to actually, now we're up and running, to actually focus on where the synergies might be. So I'm certainly excited by the progress I've seen today with the other nodes. Next slide. So this is the team. So I'm joined by Pleasantine Mill in Edinburgh, who's a cellular biologist, and Albert Ong in Sheffield, who's an adult nephrologist, and Eamon Sheridan, who's a clinical geneticist in Leeds. So this is the team. We have a research manager, Phil Hall, I think is on the call. We changed manager actually a few months ago from Angela to Phil, but there was like a seamless transition, so I was very pleased with that. And also we had a slight upset in our extremely good patient advocate died earlier in the year, so we've had some juggling around that. But the shoes have been filled by a lady called Audrey Hughes, who's a patient advocate and patient expert, and she's been instrumental. Next slide. So our vision was to say, you know, these are rare and not so rare sort of the group of diseases called renal ciliopathies. It includes ADPKD. So people argue about ADPKD, autosomal dominant polycystic kidney disease, whether it's truly rare, but actually it probably is. It's around 1 in 2,000. But we're including that with the ultra and nano rare syndromic forms of cystic kidney disease like Joubert syndrome, Jeune syndrome, Bardet-Biedl syndrome, these things that you might have heard of. But we wanted to develop this cohort, get a trial ready sort of situation where we could do these rare trials and less rare trials and build partnerships, particularly with this industry and technologies like antisense oligonucleotides and small molecules, nonsense read-through compounds, etc. So we've made some progress there. Next slide. So as I alluded to, we've got our exemplar disease, which is autosomal dominant polycystic kidney disease, and we're hoping to sort of learn from the clinical trial space and the sort of the registries and databases with this less common disease, more common disease than the less common ones as the syndromic forms of recessive kidney diseases. But actually in all disease areas, whether it's ADPKD or the recessive forms, there is unmet need. There is no drugs that switch off the disease. There's a couple of drugs that might slow things down, but there's certainly no cures. And so this is a sort of large unmet need. Next slide. So yeah, I don't need to dwell on these, but I think what we've done really well over the last year is build community. And I think this funding has allowed us to do this. So we're very much part of the renal community. Cystic kidney disease is a big burden for kidney failure, and chronic kidney disease patients accounts for around 10% of all those types of patients. 10% of all transplant patients have a cystic kidney disease. So it's big groups of patients, and we're helped actually by 2 major charities. There's PKD Charity and Ciliopathy Alliance. And so I think we've done well to enhance and build on these communities that were up and running at the start. Next slide. And as I mentioned, these partners initially were slightly aspirational, but now are coming into fruition. So, for example, Colin Johnson in Leeds is sending us murine models of a certain ciliopathy that we're working with. But I'd like to realize stronger links with NATA and with the congenital anomalies cluster, etc., as listed here, and become international as well as national. Next slide. So I mentioned we've been talking a lot about registries today, but actually one of the strengths of our application was that we're building on a really good registry called RDR, which is UK's sort of national rare disease registry for kidney diseases. And there are several thousand patients with ciliopathies already recruited. Next slide. And actually, this illustrates the power of a registry. So we have longitudinal data going back sometimes for 30 years on patients with polycystic kidney disease of various sorts. So the majority of these patients, as I mentioned, have ADPKD, so there's nearly 8,000 patients in the database. You can see median follow-up is 14 years, and it becomes a very, very powerful tool to look at survival of kidney and survival of patients, and sort of health demographics of these patients. For the recessive diseases, there's, as you imagine, fewer patients, but we've really made an effort to enrich both the already recruited patients in terms of data enrichment and genomic data, and recruitment in terms of the recessive disorders, so that now we can really boost this number. And like any registry, it has its enthusiasts, and so we know geographically that some centres recruit like crazy and some centres don't. So we're trying to incentivize recruitment. But this is a portfolio database, and so there is incentivization for recruitment for local reading units to get their patients on here. Next slide. People have been talking about the projects. I don't really want to dwell on this, but we do, as all the nodes, have 3 projects. One is based in Sheffield, which is more clinical and based around the imaging and quantification of sort of kidney size with genetic information. The project in Edinburgh is for deep phenotyping and biomarkers of disease. And then the project in Newcastle is about using antisense oligonucleotides as a potential therapy for some of these rarer ciliopathies. And the community building, I've mentioned before that the patients have been key to help us bring this together. And the other charity I've not mentioned so far is BBS UK, which is the Bardet-Biedl Syndrome UK, which is a very active disease area and one of these nationally commissioned services for patients. And so they do collect very good data. Next slide. So locally, I've managed to, as well as RDR being a portfolio study, I've managed to get now ethical approval to collect urine samples from patients with cystic kidney disease. I had this already established, but the advice I got was to reapply under the CILIAREN umbrella to get this as a portfolio recognized study. And so I've jumped through all the hoops to do that, and now it's adopted as both a portfolio study and ethically approved. And the idea here is any patient that provides a urine sample, we can grow cells that are washed out from the kidney, and that provides like a personalized model. And thinking about other diseases like tuberous sclerosis, we have the ability to look for mosaicism and other things once we've cultured the cells and grown them, and the downstream applications are really quite powerful. Next slide. So yeah, keep going. And next one. And next one. So you might be wondering what the term ciliopathies is. So the ciliopathy is this, the cilia is this antennae that sticks up like a flagpole on the apical surface of nearly all epithelial cells. And these cilia are decorated with proteins. And so any protein that's encoded by a gene that's mutated gives cystic kidney disease tends to express itself on the primary cilium. And so these are called sister genes now, because the phenotype is cystic kidney disease. So polycystin 1, polycystin 2, and virtually every other protein involved in cystic kidney disease is expressed on the primary cilium. And so this is our focus of both phenotyping and sort of our therapeutic readout in terms of can we rescue the primary cilium. Next slide. So beyond the kidney, the cystic kidney disease does have extrarenal manifestations, largely liver fibrosis and liver cysts, and there's overlap with other nodes in these disease pathways, but also eye disease, brain disease, and skeletal malformations. And so these are truly multi-system disorders in many patients. Next slide. Of course, looking at urine isn't trendy right now, but it's an ancient practice, and it's actually the oldest biochemical test or laboratory test. If you have the new technologies of multiomics allow us to powerfully use urine as a sort of dynamic biomarker, what Dave was mentioning. So we hope to use urine-derived cells as a readout for our clinical studies in real time to determine effects on genes, but also effects on sort of omics disease signatures, like are we fixing the transcriptome of a disease pathway. Next slide. Okay, and so I've applied to 4 ciliopathies to be recognized as a rare disease collaborative network, and modeling sort of what's been done for tuberous sclerosis. We have these multidisciplinary clinics that run in Newcastle, also in Cambridge and Manchester, that see whole families with cystic kidney diseases and related syndromic forms to A, establish a genetic diagnosis, and B, to manage them, and C, to provide research samples. So that's our sort of route in terms of getting patients on board with CILIAREN. Next slide. And these are some of the families that have already taken part. Next slide. So yeah, just to drive home the point that it's a very powerful technique of seeing a patient in clinic, asking for a urine sample, consenting them, taking it to the lab, spinning the cells down and growing them. And so the downstream applications of these are huge. And so we can make 3D models, do immunofluorescence studies, proteomics, transcriptomics, get DNA for long read sequencing and all the rest of it. So very, very powerful. Next slide. And the next one. So I mentioned public and patient involvement. So Audrey Hughes has established now a patient sort of strategy group for us with representation from various forms of cystic kidney disease. We're particularly keen not to have this just as a autosomal dominant sort of patient group, but we've recruited patients and patient relatives and patient parents to have a broad representation of all the syndromic forms, which I think is going to be really useful in going forward. Next slide. And we've learned from ADPKD. They did a lot of work with the James Lind Alliance to determine research priorities. And the top priority is to find a treatment for us. But actually, some of these priorities were really interesting and unexpected, and people really wanted to focus on things that doctors tend to ignore, like pain and pregnancy and lifestyle interventions, rather than a drug to fix the problem. So again, we're learning from ADPKD and applying this knowledge to the rarer forms. Next slide. I'm almost at the end. So yeah, what have the easy wins been, and what's the future direction? So the RDR recruitment, I said, is excellent. 8,000 patients already on our database and growing rapidly due to research nurses enriching and recruiting patients. My colleague in the pediatric nephrology world, Louise Oney in Liverpool, won the Life Arc Award to develop a kidney ecosystem. So this is based on pediatric kidney diseases, but all these kids will transition to adult services, and so we're closely working with this Life Arc kidney ecosystem to ensure that renal ciliopathies are represented. I've mentioned public and patient involvement. I've applied for ciliopathies to be part of this rare disease collaborative network. I'm not sure how the applications are judged. There's no money that comes with this network status, but just a badging thing. But I think, as the mTOR pathway has shown, it's really powerful, and it then becomes a way into the rare disease registry that NHS England is setting up. And there is funding from Kidney Research UK, and they're looking to establish like a virtual sort of centre for cystic kidney disease. So I think we're advertising ourselves as the main player to take part in this kidney research bid. Okay, next slide. The challenges, yeah, we were working now quite closely with NCARS to thrash out the sort of problems of getting a researcher access to the database. What we really wanted was actually a test data set that was anonymized, so that we could see how powerful or not their data was and how we could interrogate and integrate it. But I think, hopefully with help from the hub, we'll get this ironed out. Next slide. So yeah, these were our ambitions, and I think we have created a good community. We're leading a step change in thinking about cystic kidney diseases as a whole in terms of genomic diagnosis, management, and clinical trials. We're involving the rare and ultra rare. We're very much about translational therapies, but space for everyone to jump on board. Next slide. And I think that was at the end. So thanks very much.
Thank you, John. That was great, and thank you to everyone for sticking to time. Anything finally from you, Anna or Vicky?
No, nothing else. So thank you for all the presentations, which were great.
Yeah, I echo that completely. So thanks everybody, and we'll see you all in due course. I hope the rest of the day is good.
Thanks everybody. Bye.
Thank you.
Thank you. Thanks.
Bye.
Bye-bye.
Bye.